We're getting now ever closer to use our PCR machine to amplify the DNA that we've extracted along the process through Mary. So on the inside here, we've got a grid, which you probably will see in a moment, and to make sure that we can identify our samples, just a simple little scheme such as that, and we can identify in situation A1, we've got student X, student W, student whatever we want to call them. So I just use something like that to keep a track of which students have got their DNA in their particular little boxes. It relies upon, as you probably are aware, a series of changes in temperature for the DNA to unzip, to build again and to zip up, and then we go through a complete series of cycles, aren't we, about to do that? So would you like to tell us about the sensitivity of the tubes, the nature of the tubes, and a little bit more about the programming that we need to put into our PCR machine?
Okay. So the program that we're going to use today, you can see on the front of the machine here, and we've actually inputted that ourselves because every PCR process is different.
Okay.
And the difference is we're going to use primers, small pieces of DNA, and they attach to our chromosome at different temperatures. So every PCR process will depend.
So that depends on the protocol that you're using.
That's right.
Yeah,
so
okay.
you follow whatever the protocol tells you, and you put the instructions for the PCR in here. And it's basically a series of temperature changes, and it is this block here that will actually change temperature for precise amounts of time. And fitting into those very small cavities, we use very small plastic tubes. And
so even way smaller than our microcentrifuge tubes, we're reducing
really
down
small,
in volume all the
They're
time,
just
aren't
about
we?
0.2 mil.
Okay.
What you can see as well is that they have a very tight stopper on, and it's important that we keep them tightly stoppered before we use them to keep any foreign DNA out.
Yep.
And also after we've loaded our samples, we keep them tightly stoppered as well, because otherwise the very small volumes that we'll put in will evaporate at these temperatures.
Right, okay.
So we want to keep the temperature, we want to keep the contents inside.
Sure.
We also want to keep the contents of our tube down at the bottom where they're going to be heated and cooled and whatever. So on the lid there is a heating pad, and the heating pad heats the lid of our PCR tube so that there is no condensation on the lid. All the liquid is down the bottom of the tube where it is going to be heated. Now you can see these tubes are very tiny, however we must really make sure that they are fully labelled on the actual length of the tube and also on the lid, to make sure that we can identify our samples after the process.
Okay, can I have a go to just try and do one to see how clumsy you can be?
Not too easy.
Okay.
In case the writing rubs off, it's also very important that we write down on the grid which was our tube, so that we can identify our tube after the process.
Yeah, well, marking the lid's nigh impossible, but you can just about maybe squeeze an initial on the side. So I know that I'm going to call myself JB, and I'm going to go for second one down. So if we're going to go A2, so that's my location inside A2. And then other students clearly can come and put theirs in. Sorry for being pedantic, but is there any rhyme or reason why they maybe might need to spread out? Let's say we have 10 students, would we spread them out, or does it matter where they are? It
doesn't matter, provided we can identify where they are.
Okay.
You have to think carefully about whatever process you're going to do before you actually start on it with the class, because as you can see, some PCR machines are quite small. This is one of the smaller ones. There's just 25 wells in here.
Yeah.
So each student, you're basically looking at, in your class, each student would be able to provide one sample. But
in a class of? 25?
In a class of 25.
Okay.
It might be sensible to have additionally, though, control samples, which we'll talk about later.
Okay.
So you would need to manage the class so that you could fit the control samples in.
So
as well.
we'd need to backtrack then from a maximum of 25,
Yeah.
the appropriate number of controls, and therefore the number of students that can input at any one particular time.
Yeah.
Okay, thank you.